All right, today we're pulling off a microscopic heist. It's a scientific quest to get our hands on the most important molecule of life in its absolute purest form, DNA. So let's dive right into our mission briefing.
This quote really hits the nail on the head. Getting a clean, pure sample of DNA isn't just some random lab exercise. It is the absolute bedrock, the starting point for pretty much every major breakthrough we've seen in modern biology.
So what exactly is our target? Well, it's DNA, the famous double helix. And that twisted ladder shape isn't just for show. That structure is exactly what makes it so incredibly stable. It's what lets it faithfully pass down the blueprint of life generation after generation.
So why do we go to all this trouble? Well, think about it. Without a pure sample, none of this incredible science is even possible. It'd be like trying to read a recipe book that's just been completely drenched in mud. You gotta clean it off first. From mapping the human genome to catching criminals with forensics, every single one of these starts with a high-quality, intact strand of DNA.
Okay, so how do scientists actually do it? This isn't some chaotic smash and grab. No, it's a very precise, four-stage operation. This is our mission briefing.
It really boils down to 4 key steps. First, you gotta crack the cell open. That's lysis. Then, you purify the DNA from all the other cellular gunk. After that, you concentrate your sample to get just the good stuff. And finally, you run a quality check to make sure you've actually got what you came for. Let's break down each one. All right, first objective, we have to breach the cell's defenses. You can think of every cell as this tiny little fortress, and our treasure, the DNA, is locked away deep inside. We need to get in. And this is where the specialized toolkit comes in. You don't just use a sledgehammer for everything. No, you need the right key for the right lock. For bacteria, you've got lysozyme, which just chews right through their unique cell wall. For plants, you've got cellulase. And for pretty much all cells, you use a detergent, kind of like soap, to dissolve their fatty outer membrane. Oh, and EDTA, that's like a chemical crowbar. It yanks out these key metal ions that hold the wall together, making the whole thing unstable. And this is so important. You can't use the same strategy for every cell. It's a game of precision. A good scientist has to know their target inside and out and choose the perfect key to unlock its specific fortress. This chart just lays it out so clearly, right? Bacteria have this tough peptidoglycan wall, so boom, they need lysozyme. Plants have that rigid cellulose wall, so you bring in the cellulase. But what about animal cells? Well, they're actually the easiest target. They don't have a wall at all, just a membrane that a simple detergent can pop right open. Okay, we're in. The cell is officially broken open and the DNA is floating free. But now, it's a mess. It's mixed in with everything else inside the cell, a whole soup of debris. So now it's time for the getaway. We gotta separate our treasure from the trash. And the biggest piece of trash, the number one contaminant we have to get rid of, is protein. The cell is just packed with proteins. And right now, they're all tangled up with our DNA sample. So how in the world do we get them off? Well, we use this powerful old-school technique called phenol chloroform extraction. Seriously, for decades, this was the absolute workhorse of molecular biology. The basic idea is simple. You use these organic solvents that proteins just can't stand. You mix them in, and the proteins all clump together and fall out of the solution, leaving our beautiful DNA right where we want it. And the process itself is actually surprisingly simple. You take your cell soup, you mix it with the phenol and chloroform, and then you stick it in a centrifuge and spin it like super fast. And just like that, the whole thing separates into these nice, neat layers. All you have to do is carefully suck out that top layer. And this is what you see. The DNA, it's perfectly happy in that watery top layer, so it stays right there. All those unwanted proteins, they get trapped in this gross white clumpy layer right in the middle. And those heavy organic solvents, they sink straight to the bottom. So our prize, our treasure, is right there on top, waiting for us. But wait, hold on. Our mission is not quite over yet. We've managed to get rid of all the protein, but DNA's chemical cousin, RNA, is still hanging out in our sample. We gotta get rid of that too. So for this final delicate cleanup step, we bring out a molecular scalpel. It's an enzyme called ribonuclease, or just RNase for short. And this thing is incredibly specific. It's like a guided missile that seeks out and destroys only RNA molecules, leaving our precious DNA completely untouched. It is the perfect tool for the job. So, the extraction is done. But did it work? Was it successful? Now we have to play detective and actually verify the purity of our sample. It's time to check our work. To do this, scientists use a really cool machine called a spectrophotometer. Basically, it shines light through the DNA sample and measures exactly how much light gets absorbed at different wavelengths. See, DNA, protein, and other chemicals, they all absorb light differently. They each have a unique signature. And when it comes to pure DNA, there is one magic number that every scientist is looking for, and that number is 1.8. This chart explains everything. What scientists do is compare how much light is absorbed at 260 nanometers, that's where DNA and RNA really shine, to the light absorbed at 280 nanometers, which is the peak for proteins. If that ratio is a perfect 1.8, then congratulations, you've got pure DNA. If it's higher, you still got some RNA in there. And if it's lower, that means you've got leftover protein. It's an incredibly elegant quality check. And there's also a second crucial check they run, the A260 to A230 ratio. This one is looking for different kinds of chemical leftovers from the extraction process itself, things like salts or maybe some of that phenol, which happen to absorb light really strongly at 230 nanometers. For a truly, truly clean sample, this ratio needs to be well above 1.5. So, we breached the cell, we fought off all the contaminants, and we verified our prize. We officially have pure DNA. So let's circle all the way back to the beginning and remember why this whole meticulous heist matters so much. Because with this pure sample, the possibilities are just about limitless. We can engineer life-saving medicines. We can diagnose genetic diseases before they even start. We can identify a criminal from a single cell. We can map the entire genome of a species, and we can truly, finally, begin to understand the source code that runs every living thing on this planet. And that, of course, leaves us with a pretty profound question. This fundamental technique has given us the power to read and more, to write the blueprint of life itself. So the only real question left is, what do we want to build next?